research is a phylogenetic review on monkeypox virus. So it was made by my mentor, Barada Khat, a graduate student at the University of Calgary, and me. Okay, so first of all, I want to introduce monkeypox, and it's like the main reason we did this analysis. First of all, because I think most people here know that it has been declared a public health crisis, like since May of 2022, because of all the increment in, well, confirmed cases of this disease. So, what is the monkeypox virus? It is the virus that it is responsible for monkeypox, a rare zoonotic disease that exhibits symptoms to smallpox. So this virus has mainly two strains. One is the Congo Basin clade, and the other one is the West Africa clade. The Congo Basin clade has been reported to have a bigger fatality average rate than the West African clade. So this is also a member of the Gardapoxvirinae subgenera family, which is also a member of the Poxviridae family of the Orthopox genus of viruses. This virus has a brick shape and it has a genome of approximately 130 to 380 kilobase pairs approximately, and a size about to 200 and 200 nanometers. So it is pretty small, and that's why it can be like so problematic for nowadays health. So what about the history of monkeypox? First, this is a virus that was first discovered in animals, mainly in Singaporean monkeys that were traveling from Singapore to Copenhagen in 1958. So there was like this investigator in the crew of transporting animals, and he saw that one of the monkeys was infected with something similar to smallpox. So he decided to take him to the vet and, you know, like research the disease, and they found that it was like a novel disease, right? So that's why they decided to name it monkeypox. In 1970s, the first 6 human infections of monkeypox were reported in Central and West Africa. Actually, the first infection was reported in the Democratic Republic of Congo in a 10-year-old child. So that was like very sad for me to hear. And how did he get infected? It is known to be an animal to human transmission, but people, well, in the literature reviews, they don't say like where is possible that he got the disease. So more recently, there had been like more outbreaks outside this region of Central and West Africa. Actually, 10 out of the 35 monkeypox outbreaks outside of this region, which is considered the region of origin because it was the first region where human cases were reported, have occurred since 2010. So this means that like the majority of the cases have occurred like the half of the time after the disease was discovered. So this monkeypox disease was declared an epidemic in 2017 because a Nigerian outbreak that was very strong and that caused like many concerns on health in Central Africa. In 2022, Europe has been presented as the epicenter of a new outbreak, specifically the UK. And as of July of 2022, more than 21,000 cases have been reported in 78 countries. But like I recently checked, and as of August 2022, more than 41 cases have been reported in 94 countries. So this is alarming because cases have doubled in just 1 month. And well, as you can see, like of the 94 countries, only 7 countries had previously reported monkeypox as an endemic disease. So this means that most of these outbreaks are of new origin, and they have been transmitted because of human-human interaction and not animal-to-human interaction. So my project is about mainly phylogenies. So phylogenies are trees explaining the genetic sequences evolution and also protein sequences evolution through statistical and computational methods. They are mainly used to identify the evolutionary divergences and differences between like organisms among one clade. So one clade is a taxonomical group, and they are of great importance, principally studying virus, because we can generate data about like the elucidation of the processes of the viruses with comparison, right? For example, we know that smallpox is caused by a virus called variola virus. However, I don't know if you knew that the vaccine that it is used for smallpox is actually done with another virus called vaccinia virus. So how do people knew that vaccinia virus was going to work against variola? Well, this is because scientists did like different studies, and they with this phylogenetic tree saw that these two viruses were too close to each other, that they must have like the same molecular and cellular mechanisms of infection and of replication, and therefore as vaccinia viruses had like more characteristics that made it easier to manipulate for humans, they chose that for the vaccine. So the objectives mainly of my projects are to find the connections between reported literature phylogenetic analysis of the monkeypox virus and phylogenies made by bioinformatics tools available for public use. Also identify other virus species with similar mechanisms to help with research, and discuss the utility of phylogenies in other research areas like immunology and epidemiology. So for the previous research has not been made like specifically on the monkeypox virus, but on bigger clades such as Cordopox birinae. So Cordopox birinae, it's like the subfamily of this virus, right? What Cordopox birinae has is virus that infect all chordate animals, meaning all animals that develop a vertebrate column. So this family is actually very big, and has had like so many research on phylogenies using principally single analog proteins and complete genome. So single analog proteins are like phylogenies are like evolutionary trees made on only one gene, right? Or one proteic product of that gene. So I found like mainly three articles. One of 2006 that it was made using the pox virus clade, which is like the family of Cordyceps virinae. So in this family, there have been identified 49 conserved genes. So this means that these genes are essential for the virus replication cycle. And if one of these genes is missing, then the cycle cannot thrive in the infection. So in this review, well, in this study, like the monkeypox virus was grouped with several others, which include cowpox, vaccinia, ectromelia, variola, rabbitpox, and camelpox. Then in 2021, another pox virus phylogeny, they used 60 sequence of pox virus, and in monkeypox grouped with vaccinia again, horsepox, variola again, tatarapox, and camelpox again. And also in a 2004 orthopox virus phylogeny, which was one of the first ones ever made, they identified 12 different core orthopox virus amino acid sequences, meaning that these proteins were very well conserved and very important for the virus cycle. And they grouped monkeypox with ectromelia, cowpox, and vaccinia, which it is not a coincidence that, well, monkeypox has always been grouped with these viruses, right? Which includes like ectromelia, vaccinia, cowpox. So my methods for doing the phylogenetic tree was at first we choose the orthopox genomes and the protein sequences from the NCVI database. So the first thing is that as our main research organism is the monkeypox virus, we selected first this sequence. So we choose monkeypox virus Cyrus strain 96I16, with has like several reference sequences. A reference sequences is a sequence that has been like reviewed by the NCBA to be like the official sequence of an organism. So we use the reference sequence, and also we use the reference sequences for genes I7L, F8L, and E8R. We use these genes because they form part of the core genes, which are into the group of the 49 genes conserved in pox virus family, and also because they are essential for the virus protein cycles, and they participate in different parts of the process. So for example, the I7L virus participates in the morphogenesis of the virus. So that means the assembly of the capsid and the envelopes that the virus has. The F8L that is involved in replication of the nucleic acids, and the E8R that participates in the transcription of the DNA material of the virus. Then we did a blast and alignment for genome phylogeny. So for those that are not familiar with the NCBI database tools, the blast is a multiple alignment processing tool in which you put like a sequence. This sequence can be a known sequence, or it could be a sequence that you get on your own from your lab, and then the database will pair that sequence with all the other sequence above like a certain similarity rate, and then will give you the alignment and some trees regarding like the information obtained from that multiple alignment, right? So multiple alignment, what it means is that we put like all the sequences like in the same order and in the same format, and we try to look all the regions that have like the same bases or similar bases, or not only like nucleic acid bases, but also the same amino acids, and we draw the percentage of the similarities and the differences between them. So first we did a BLASTn alignment. BLASTn is for only nucleic acid sequences. So that one was for the genome, and then a BLASTp, which is for proteins. So that one was the one that we use for the single genes. So in this case, we use for the genes the protein sequence and not the nucleic acid sequence, because the protein sequence doesn't have like as much as neutral mutations. So neutral mutations are those that are highlighted in the DNA, but they don't have like any result in the proteins. Then after that, we did like the multiple alignment. So the algorithms that the NCBI uses is cluster W. And also we did a multiple alignment on a public use software tool called MegaX, which has like the same algorithm. So we run the multiple alignment on both sides. And then we did the phylogenetic analysis. So this is like the interface of the MegaX software where we use cluster W. And as you can see, we have like the colors. The colors represent actually like the amino acids and the chemical nature. And also, if you can see here, we have several like asterisks. So this means that the sequence is like, well, it's like conserved in all of the organisms that you study. So for the results, these are the resulting phylogenetic trees. So first we have the tree that resulted from the genome. Then we have the tree from the F8L sequence, the tree from the I7L sequence, and the tree for the I8R sequence. So these trees were like kind of modified, were colored modified. First, I highlighted in red like the monkeypox virus sequences. So as you see, we chose like a specific strain, which was Sire. So in some blasts, the monkeypox virus like general sequence appeared. Then we highlighted in blue like all the other organisms that were previously reported in literature to be grouped with monkeypox, and we grouped in green and organisms that were also grouped into those groups, like within the monkeypox and the literature organisms mentioned, but that they were not previously mentioned, right? So in most cases, like we find like all the viruses grouped within the same subgroups, and but however there is like some difference, mainly in length branching. So length branching, what it means, it's like the number of difference between one strain of a virus and the other one. So in this case, like for example, it means that the monkeypox virus has had like more evolutionary changes. So like it appeared like more recently in the history of time than, for example, the monkeypox virus that has like a shorter difference, right? And also, as you can see, like here, the knots represent like the grouping of the virus. So here, monkeypox is grouped with vaccinia, as same as in the upper tree, in which monkeypox is also grouped with vaccinia. And we have like sister groups, which are the groups that are like uh continuously to the others, right? So for example, here this group containing variola, camelpox, and tetanus is a sister group of vaccinia and monkeypox group. So this is how we read phylogenetic trees. And for our discussion, we could see that the results differ among the different trees because each sequence mainly represents different variability and evolutionary patterns and processes. So what this means is that a gene is not going to evolve at the same rate nor at the same method as a complete genome or as another gene that has a different function. But however, similarities were also found among trees because all of these organisms share common ancestors. So we have that vaccinia viruses tends to be closer to monkeypox virus, and variola, camelpox, and tetanus usually are represented within the same group, which is a sister group of the subclade, including the monkeypox virus, and ictromelia virus is usually placed as an external group. So also this is important because therefore we can root the phylogenetic tree between ectromelia and the other viruses in order to get like a more accurate representation of history. And also, well, genome analysis considers the organism as a whole, meaning they get to have information about all the mutation rates and all the evolutionary processes that the organism has been through. So this report also had its limitations. First of all, complete genome analysis haven't been like 100% completed because the extreme sequences of all the viruses are extremely divergent. So the algorithms developed until now have like many problems to align those sequences. So what most scientists do and what we do is to slice only like one part of the central genome and then that submit it to the multiple alignment. Then there are also a lot of reports on the phylogenies because this virus is of recent human interest, so it hasn't been like a lot investigation done on it. And the methods used by each phylogeny are different. So for example, I've seen that mostly the algorithms made to process the multiple alignment information into the phylogenetic tree are different. So ones use like neighboring joining trees, one use maximum parsimony, one use maximum likelihood, and that causes a problem at the time of uniforming investigation. And also, like most of trees are rootless, so we haven't find like a common ancestor for all this clade. So this means that we can only measure like the differences, but not an actually evolutionary history. And well, we also made like a little epidemiology review. So obtaining information about the relatedness of this virus, but not only like this group of viruses, but all viruses and their taxonomic groups allow scientists to draw inferences and conclusions about unknown characteristics in this virus. For example, the monkeypox virus is that doesn't have hasn't been researched on the protein that allows the enter of the virus to the cell. For example, for coronavirus, we know that it is like the spike protein, but for monkeypox virus, we don't know what protein is nor how the process works. However, because of its relatedness to variola, we now have like a more close idea of how can this work. Also, in the Democratic Republic of Congo, like cases have aroused like very quickly. For example, in the decades of the 2000s, there were 10,000 cases reported. Then this number almost doubled in the next decade, and now like we have like 16,000 cases, but just in 1 year, which means that this number will probably make 3 times itself at the end of the decade. Also, in the last 50 years, except for 2022, only 14 countries had reported monkeypox. As of today, 94 countries have reported monkeypox. Also, the increase of cases has been attributed to waning immunity, which means that before we have this population immunity granted to the vaccine, which prevented disease of the smallpox virus. However, as that vaccination ceased on the 1980s, then immunity has been decreased among the populations. Also, monkeypox, in our opinion, must have a status of a pandemic because a pandemic is defined as an epidemic with international spread and also like high cases. Very high cases. I think that monkeypox, at least for the international spread, it like accomplishes that status. So I think that people and mostly the health governmental offices must start to review like their policies about how to manage monkeypox viruses among their countries. And also, we must continue to study Cordopox viridae viruses because it has been useful for epidemiological analysis. And as far as we know, most of these viruses can infect humans. So if we have had like smallpox pandemics and now monkeypox pandemics, well, who knows what the next Cordyceps viridae virus will cause a pandemic. And for conclusions, well, our phylogenies were actually very similar to those reported in literature. Monkeypox has alarmingly increased all over the world. And well, we should continue doing phylogenetic analysis. And for recommendations for future projects, we should elaborate more on the essential issues that prevent the population to be prepared for, and the scientific community to be prepared for future pandemics. For example, we should investigate more about the immunity aspects of the virus. For example, what is its mechanism to evade immunity, like the immunological processes from our bodies, and also the virus structure, because as far as we know, like only two of the main core genes have a study on their structure and the protein products. So we must hurry in that part if we want to develop like stronger vaccines and stronger treatments. So these are my references, and I want to give a big shout out to my mentor, Barbara Cott, which made like this project possible and which introduced me like to all of these of processes to make phylogenies, and also to the Talaria Summer Institute to make this research possible. Thank you.